Hello everyone. Today's topic is Histopathology and Drug Development Series 2. We're focusing on sample processing and H&E stain. This is the disclaimer for this presentation. Histopathology assays essentially are lab testings that focuses on human or animal tissue processed into thin sections for applications of various stains. Most commonly, we use fresh frozen or formalin-fixed paraffin-embedded tissue blocks or slides. Histopathology studies has been indispensable tool for development of medicine and biology or diagnostics for more than 100 years. Basic histology specimen processing and stains such as H&E are more sophisticated as it seems. For example, this is a H&E stain on two different tissue. The one on the left side is nicely done. You have all the cell and tissue morphology and structure. However, on the right side, it's a poorly done H&E stain where you can probably not get much of information out of it. As many other laboratory tests, multiple factors in different stages or phases impact the final results and quality. We divide them into pre-analytical phase, analytical, and post-analytical phase. In pre-analytical phase, factors impacting histopathology assay qualities include some of the important ones listed here. Cold ischemic time essentially is the time period between when you take the tissue out of the living body, being a research animal or human subject, to the time point you put them into fixatives. And we recommend it to be less than 30 minutes. Reason being, when the tissue left the live body, they have the blood supply, nutrition, and everything cut off. And the process of hypoxia or even autolysis starts right away. Some of the biomarkers you're trying to look at in those tissue might also starting to disintegrate. So that's why it's very important to put them in fixatives as soon as possible. The next one is so-called prefixation grossing and fixation time. Prefixation grossing essentially means that if you have a larger chunk of tissue, or on every dimension the size is more than 10 or 20 millimeters, you might consider of doing some further sectioning or slicing before that you put them into fixative. Reason being that the speed of fixatives, in this case normally 10% neutral buffered formalin, penetrates tissue at a speed of approximately 1 millimeter per hour. Therefore, if your tissue is too big, or there's certain fibrous capsules on top of the entire organ, such as kidneys, you want to cut the section into thinner slices before you put them into fixatives, so that the fixatives can reach the tissue core and center, ensuring complete and thorough fixation. In terms of fixation time, many guidelines have given that you should fix the tissue between 6 hours to 48 hours. Of course, you don't want to underfix the tissue with less than 6 hours, but overfixing is as detrimental to the tissue as underfixing. Some of the guidelines says 72 hours is probably fine as well, but we really don't recommend fixation more than 48 hours, especially in smaller tissue size. However, some tissue types, such as adipose tissue or neural tissue, are known to be very hard to fix. Therefore, in these kind of tissue types, you might appropriately expand the fixation time a little bit longer compared to other tissue types. In terms of analytical phase, many factors can also impact the assay quality. But today I want to bring up two important things that might be often ignored in assay processing, in assay performance. The first one is so-called sectioning thickness. So normally for histology sections, especially for FFPE, a regular section size or thickness is 4 to 5 microns, and for frozens, we can do 5 to 10 microns. In order to ensure that the staining, downstream staining you'll be applying to these sections later generates reliable and accurate results, especially for some semi-quantitative or quantitative studies, you want to make sure the section thickness is consistent or at the desired thickness you set on the microtome. And we'll give you some examples later of how important this is. And another factor is the inconsistency caused by manual staining or lack of QC on automatic staining machine. We're not going to get into too much details on that because it's quite easy to understand. For post-analytical factors, there's also some details you want to pay attention to. For example, when you're applying cover slips to the glass slides, you want to avoid air bubbles. There's certain things you can do to avoid air bubbles. And in a lot of labs these days, digital pathology is a mainstream. And then you want to scan the glass slides into whole slide images. And there's also many technical details you want to pay attention to, but we won't be presenting or discussing these in today's presentation. Finally, human factor and environmental factors. And these are the human factors are easy to understand, that different operators on the same assay might create variances, and there are a lot of procedures and workflows you can control and limit those variances effectively. The environmental factors essentially refers to the temperature and humidity of the laboratory, of the histo lab. In most of the cases, the humidity is more important because that has a big impact, especially on the FFPE sectioning. Okay, so we have given a brief overview of histopathology specimen processing. Now we want to bring up some tips and tricks for some commonly seen problem in histo lab. Hopefully those are helpful to you. First one, we'll be talking about brittle tissue blocks. So essentially what it means is that when we're cutting FFPE blocks, you can see from the video shown here that some of the blocks just won't give you very smooth and intact tissue. The tissue tends to fall apart, creating all those cracks and holes. And also, it almost feels like you're cutting through a cookie instead of a tissue. And how to resolve this? We have a very simple trick, which is taking the block off of the microtome and putting it face down into ice water bath. You can leave it there for like 20 or 30 minutes so that the block can soak up the moisture enough to soften the block. And if the block is extremely dry, you can also leave it there for about an hour or two, and you don't have to worry about that having any negative impact on the following stains. So as you can see in this video, we have soaked this exact same block we shown in the previous video in the ice water bath, and now it's giving you very nice continuous ribbons with intact tissue in it. The second thing is also very important, is the maintaining the section thickness consistently. When we're cutting FFPE blocks on a microtome, we always set the tissue thickness to a number, a desired number. Normally it's 4 or 5 microns. Sometimes people like even thinner, like 3 microns. The thinner the section is, the harder to keep the integrity. But also you have to pay attention whether you're really getting 3 or 4 micron sections off of the microtome. And what we have seen is that whenever you stop sectioning a block, maybe you want to take a break, or you're just taking the first ribbon into the water bath and coming back to the block and start the next ribbon, right? The first section coming off from the next ribbon is normally much thicker than the sectioning, than the sections coming after that in the same ribbon. As shown in this video, you can see we just started a new ribbon sectioning, and then you can see the first section coming off of this ribbon is actually quite thicker compared to the next one and the rest of the sections from the same ribbon. And this tells us that you probably want to avoid using that first section for any of your important stains, because you're not really sure whether that's coming off as your desired section thickness. We can put this into the water bath, and also the rest of the ribbon. You can see immediately already that the transparency of the sections starting from the second one is much more transparent compared to the first section, meaning that thickness is different. And this has an impact on your staining results. So here's the example. We did the H&E stain on those sections shown in the previous video. On the left side is the first section, which is thicker than the rest of the other sections. On the right side is the second section after the first one. And you can already see that H&E stain is much stronger on the left side, which is the thicker section. So this is an important matter if you are actually doing critical semi-quantitative or even quantitative staining or assays down the road. Applies to other stains as well. And that for the lower panel, you can see it's a liver block with IHC stain for the same marker under the same procedure, same run. And the thicker section on the left side shows much stronger signal than the one on the right side, which is the proper thickness. Okay, we can switch gear to the frozen section side. For frozen sections, one of the most common issue we encounter is again the brittleness of the block or the tissue. And there's a term for that. It's called a shattering artifact. Essentially, as you can see in the video, the sections coming out of this frozen block tends to crack a lot, making a lot of holes. And this is most likely caused due to the block has been left in the chamber for too long, so it's too cold. So to resolve this, all you need to do is just put your thumb on the surface of that block and leave it there for a couple of seconds or 10-20 seconds, and then you do the section again, because your thumb increased the temperature on the cold and brittle section, and now the sectioning come off of the block is much more intact, and you can observe the full tissue morphology without suffering from any cracks or holes made. Okay, hopefully those few tips and tricks could be helpful to you. And let's summarize the key take-home messages. Tissue collection and fixation is critical for high-quality histology studies. Details in sectioning also impacts staining quality and consistency. And brittle FFPE sections could be saved with ice water bath. And this thumb pressing technique can save you from shattering artifacts in frozen sections. And that's all for today. Please reach out to us if you have any further questions at this email address. Histopathology is becoming more and more critical for drug development and translational research, especially human specimens under a clear cap environment. iCura team has extensive experience and expertise in this area. However, we'd like to hear from our audiences for any feedbacks and suggestions to improve our service to the next level. Thank you all so much.